Thank you very much, the organizers, for giving me the opportunity to talk about our work. I should mention that the primary stages of this work were performed at the lab of Professor Joanne Corey, where I did my postdoc at the Salk Institute, and later at the University of Glasgow in my own lab. So I'm really interested in how signals integrate in the nucleus, and in particular I'm interested in how environmental stimuli such as light integrate in nuclear photobodies. So this is just to remind you that light is not only essential for energy production and photosynthesis, but also it regulates a number of developmental processes in plants from the very early stages, such as during seed germination, deethylation, or photomorphogenesis. Also, it helps the plants to compete with the neighboring plants, and also to synthesize photoprotectants. And in addition, for major developmental transitions such as flowering time. So I have always been fascinated by light signaling and plant photoreceptors, especially because plants have developed a very sophisticated family of photoreceptors, ranging from UVB receptors such as UVR8 to blue light receptors such as the cryptochromes and the phototropins, as well as the red and far red light receptors, the phytochromes, and also the Zeitlupe family of photoreceptors that are involved in circadian rhythms. Okay, and although the majority of all these photoreceptors, with the exception of phototropins that are plasma membrane-associated proteins, are either perceiving light in the cytosol or in the nucleus, or then translocated into the nucleus and then interact with a number of signaling components and transcription factors, no one really knows how this nuclear signal integration occurs. So I'm really interested in signal integration in the nucleus, and this all started based on a non-conventional genetic screen that was performed by a previous postdoc at the Corey lab, Olivier Ludet, who is now in France at INRA. So what Olivier did is he used natural variation in order to identify novel signaling components in Arabidopsis. So he collected different Arabidopsis ecotypes from all over the world, and in particular he compared Bay and Shah ecotypes in order to identify specific loci that code for genes that are important in hypocotyl elongation. And from this screen, he identified a gene that he named TZP based on the protein domain composition for TANDEM ZINC-FINGER PLUS3 domain. So in Bay, where you have a non-functional TZP due to a premature stop codon within the +3 domain, you have shorter hypocotyls, in particular in response to blue light, whereas in Shah allele that had the wild type TZP, he saw longer hypocotyls. So he also made transgenic lines overexpressing TZP in that background, and he showed that you can get longer hypocotyls. And based on microarray analysis that he did, he showed that this is due to the fact that TZP can regulate a number of genes that are involved in growth promotion, in particular for hypocotyl elongation. So this is when I started the project, and I wanted to find out how TZP, how this protein works. So at the protein level, this is the amino acid sequence of TZP. You can see that in yellow, there is an extended N-terminus sequence that is believed to be unstructured, is predicted to be unstructured. And towards the C-terminus, there is in blue 2 zinc finger domains and a +3 domain. So zinc finger domains and +3 domains are highly conserved in nature. Zinc finger domains are found in many transcription factors. They're also involved in addition to DNA, RNA binding, also to protein association, whereas +3 domains, they have been found in yeast and humans, and the proteins that contain them, preferentially they're involved in chromatin remodeling, or also they recruit splicing factors during transcription. Within the +3 domain of TZP, there is also a putative ear motif. And ear motifs have been shown in plants, in proteins such as topless, that they are recruited by these ear motifs, and they're involved in regulating gene expression, in particular, recruiting transcriptional core repressors. So, we know also from mass spec analysis that at the N-terminus of TZP protein, that there is a lot of serines that are phosphorylated, and also there's putative summoylation motifs. So we think that the N-terminus of the protein has a regulatory role. So one of the first things I did was to tag TZP with a fluorescent reporter, and also I wanted to see if I could see the same phenotype that Olivier saw in Chat, and also when he rescued the Bay mutant phenotype. So I overexpressed TZP in Colombia. And here you can see that it shows longer hypocotyls in response to blue light. And I was very interested in the fact that this protein is localized in the nucleus, and in particular in these pactate nuclear body structures. Initially, I thought that this could be an artifact of overexpression, but we used different promoters. Even with a native promoter, we could see that the protein is found in these nuclear body structures. And when I did some deletion analysis, we could see that the N-terminus of TZP is necessary and sufficient for this nuclear body formation, whereas the +3 domain or the zinc finger domains are not able to form these nuclear body patterns. Also, because it has been known that specific light signaling components, and in particular the red light receptor, phytochrome, is one of the most well-characterized proteins that form these nuclear bodies, we wanted to see if TZP nuclear bodies form in particular in response to specific wavelengths of light. And here you can see that TZP forms nuclear bodies specifically in response to red light. And as a control, we use coilin, which forms constitutively nuclear bodies irrespective of what light wavelength we use. And these nuclear bodies, they follow a diurnal pattern. So we have the maximum amount of nuclear bodies during the day and the minimum during the night. And this only happens in response to red light. So if we substitute the white light of the light-dark photoperiod with red light, we see this effect, but not when we substitute it with blue. And these nuclear bodies don't seem to have an effect at the protein level. So when you form nuclear bodies, the protein doesn't get degraded or accumulated further. We also wanted to see how mobile these nuclear bodies are. So we did some FRAP analysis. So this is fluorescence recovery after photobleaching. And when you bleach TZP in these nuclear domains, you can see that it recovers extremely fast. Whereas if you bleach TZP when it's found in the nucleoplasm, it recovers much slower, suggesting that something is actively recruiting TZP in these nuclear bodies. So what is known about nuclear bodies? They're not new structures. They were first characterized by Ramon y Cajal in 1903. And in the mammalian field, disruption of nuclear body patterns is associated with a number of diseases, aging, and cancer. And they are believed to be involved in protein stability and regulating nuclear architecture or even splicing. In terms of nuclear bodies in plants, in particular in Arabidopsis, and nuclear photobodies, as we call them, so these are the nuclear bodies that form in response to light, we know mostly from studies on phytochromes, on the red light receptors, phytochromes, that they could be act as a site where the protein, the photoactivated phytochrome, is stored. But also recent studies from Meng Chen and Freddie Barnesh's lab have shown that in response to light, we have relocation of specific loci to the periphery of the nucleus, but also light mediates the global architecture changes within the nucleus. So we wanted to see what else is found in these TZP nuclear bodies and whether TZP co-localizes with known nuclear photobodies. And here you can see that TZP and phyB co-localize in nuclear bodies and also other light signaling components such as ELF3. However, proteins that form known nuclear bodies that are not formed in response to red light, they do not co-localize with TZP. So it seems that TZP forms nuclear photobodies in particular. So it's involved in complexes of proteins that respond to light. And here, when we made transgenic plants expressing TZP in the absence of phyB, so this is in a phyB mutant background, you see that you lose this nuclear body formation, and this doesn't happen happens when you don't have phi-A. So this is a phi-B specific response, suggesting that phi-B is necessary for recruiting TZP in these nuclear bodies. Furthermore, when we used yeast to hybrid assays, we could see that TZP and phi-B interact directly in yeast, and it's the N-terminus of TZPs that is necessary and sufficient for this interaction. And this is to remind you that the N-terminus of TZP, again, is the part of the protein that is important, contains the information that is important for this nuclear body formation. We also wanted to see if we could see phi-B and TZP interacting in planta, and this is co-immunoprecipitation studies on transgenic Arabidopsis plants expressing tagged TZP with GFP, and when we pull down TZP with a GFP tag and then probe with the anti-phi-B antibody, we can see that TZP and phi-B co-purify from plant tissue, and at the same time, from a completely different study at the K lab, Miter Nussinau did some mass spec analysis using ELF3 and ELF4 as the base. So ELF3 and ELF4, they are important for light signaling and circadian regulation. And he could see that in addition to phi-B, TZP was one of the most abundant peptides that came with this complex. And in the absence of phi-B, so when Miter did these experiments in phi-B mutant plants, he could see that TZP was not part of this complex, again suggesting that phi-B is essential for recruiting TZP in these nuclear bodies and making it part of this light signaling complex. So what is happening in these TZP phi-B nuclear bodies? We took a pharmacological approach. So we use specific inhibitors to see if we can disrupt this nuclear body formation. And here you can see that in particular in response to transcriptional inhibitors, we see that nuclear bodies of TZP and also phi-B don't form, or they form a massive nuclear body. So they seem to be disrupting this nuclear body formation in response to red light. Whereas other inhibitors such as taurosporin or proteasomal inhibitors, MG132 and cyclohexamide, they don't have an effect on these nuclear bodies. And this is just a plot to show that they do not interfere with the protein expression. Well, for MG132, you see more protein. And also, when we did some in vitro studies, we could see that TZP, and in particular the +3 domain of TZP, can associate with single-stranded but not double-stranded DNA, which is again an indication of proteins that are involved in active transcription. So what is the physiological significance, though, of this TZP and phi-B interaction? Initially, we were very surprised that we couldn't see a red light or far red light specific phenotype with respect to hypocotyl elongation in response to red or far red light, because it is known that phytochrome is regulating hypocotyl elongation under red or far red light, whereas we could see what Olivier has seen, a phenotype in response to blue light. So we assume we wanted to test some other light signaling effects that are regulated for phi-B to understand what is the physiological significance of this interaction. And of course, we examined photoperiodic flowering, and this is a very oversimplified diagram of how photoreceptors such as phytochrome and cryptochrome, and also the Zeit-Lupe family of photoreceptors, they regulate the abundance or the gene expression of a major transcription factor, CONSTANS, that is responsible for inducing the expression of the flowering locus T, which then moves into the shoot apical meristem in order to initiate flowering. So here we can see that when we examine plants overexpressing TZP, we could see that TZP promotes photoperiodic flowering, in particular in response to long day photoperiodic conditions, and this is a phi-B dependent effect. And furthermore, we wanted to see if overexpression of TZP promotes the expression of FT, And here we can see that this is a developmental specific effect. So when we grow the plants for 12 days, we can see an induction of a FT gene expression, and this doesn't happen at 4 days when Olivier did his microarray analysis, suggesting that TZP may be having a developmental specific role in hypocotyl elongation during the early stages of photomorphogenesis, and later during the lifetime of the plant, it has an effect in photoperiodic flowering. Furthermore, we could see that TZP induces the expression of CONSTANS, but at a much lower level, and this is just a control showing that it has no effect on other light regulated genes such as Hi5. And we also wanted to see if TZP can associate directly with the promoter of FT. And here we can see that TZP can associate with the transcriptional start site of the FT locus. And again, this is impaired in the absence of phytochrome B. And you need the full-length protein in order to be able to associate with the FT locus. So our model so far is that phyB is essential for recruiting TZP in these nuclear photobodies, and we believe that phyB also is recruiting TZP in the promoter of FT. However, we cannot exclude the possibility that the interaction between TZP and phyB could be withdrawing phyB from targeting CONSTANS for degradation, and this is something that we are examining at the moment. However, we don't think that this is the case because TZP would not be associating with the promoter of FT in order to regulate its expression if that was the case. So in terms of what is the physiological role of TZP in wild-type plants, we believe that phyB could have a positive role in regulating or in promoting photoperiodic flowering during early in the day. For example, as ET.5, phyB may be recruiting TZP in the FT locus in order to maintain basal levels of FT gene expression in a very similar manner to what is happening with Hi5, for example. And when you overexpress TZP, obviously you tilt this balance, and that's why you promote flowering and the plants flower earlier than in wild type. So of course there is a lot of questions still to answer. We're interested to see if TZP acts as an activator of transcription, but also as a repressor. We want to see if this putative ear motif is actually having an effect in terms of TZP function. We're interested in the +3 domain, which there's no other protein in Arabidopsis that has been characterized in terms of the +3 domain function. There's 5 proteins in addition to TZP that contain +3 domains in plants. And as I mentioned, in humans and in yeast, these proteins recruit splicing factors and are involved in chromatin remodeling. And one of the big questions that remains to be answered is what is happening in these nuclear photobodies. So when we use a conventional confocal microscope, we can see these nuclear bodies, these dots basically in the nucleus. However, when we use super resolution imaging, we can see that these nuclear bodies, in particular when we examine TZP localization, they form this donut shape. When you ask people in the mammalian field, they would say that these are very similar to PML bodies in mammals. So, of course, we would be very interested to examine these structures in more detail, and in particular to see if specific gene loci are recruited in these domains when you have a collection of transcription factors and light signaling components in a very similar manner to transcription factories. So instead of having the polymerases and the transcription factors moving towards the DNA, you have specific loci that are co-regulated, moving to specific domains within the nucleus that you have all these protein signaling components. And in addition, we know that PML bodies in animals, they are associated with protein modifications and in particular summoylation. So it would be interested to investigate whether TZP is summoylated. We know that phytochrome B is summoylated, and based on mass spec analysis from the Vierstrand lab, we know that TZP is a putative summoylation it has putative stimulation motifs, but it's also found in global proteomic analysis. So it would be interesting to mutate these sites and to see if we would interfere with this nuclear body formation. And with that, I would like to thank, first of all, Professor Joanne Corey for allowing me to take this project with me in Glasgow, at the University of Glasgow for my fellowship, the BBSRC and the Wellcome Trust, and the Human Frontiers for my postdoc fellowship, and people in the lab that have contributed to this work. And thank you very much for your attention.